wearing garlands and a shirt just because Australia, if those that didn't know, in Africa, Australia won the Cricket World Cup yesterday. So that's the reason for celebration for some. But yeah, welcome to a seminar here from a former colleague of mine. So Nancy A Schellhorn comes from CSIRO in Australia, and Nancy and I used to work together in the agricultural flagship and before I joined ICRISAT. So it's a real pleasure, Nancy, to have you join or come and visit ICRISAT and talk to Harry Sharma and his team around the work that ICRISAT does in entomology and IPM and other areas. So Nancy's agreed to give us a seminar here. Nancy is a principal research scientist and leads the spatial ecology team in CSIRO in Brisbane. And they do some fantastic work for the Australian grains and cotton industries around IPM management, not just from an agricultural perspective, but also an ecosystem service perspective. So I'm sure Nancy's going to touch on much of that in her talk. So it's a real pleasure, Nancy, to welcome you here and to invite you to give a seminar. Thanks.
So can you hear me okay with this mic?
Yeah. Yes.
So Peter, thank you very much for the introduction, and thank you so much for welcoming me to ICRISAT. I've always wanted to visit, and so what a fantastic opportunity. I was in Bangalore to come up and also to spend the morning with Dr. Sharma and his lab. So about 25 years ago, I got interested in the way in which insects compete with humans for food. Now, arguably, at the time, I was probably more interested in the food angle, but then I learned about some of the fascinating aspects of insect ecology. But I was also aware of the often environmentally unacceptable ways in which we go about trying to control pests to stop them from eating our food. So I pulled this up on here, is that correct? Okay. So how are we doing that? Well, that was 25 years ago. Ork has done some work, and his quote is, despite a clear increase in pesticide use, crop losses have not significantly decreased during the last 40 years. So shown here is some work from Ork. I hope I get this right. You know, you need one finger to push 3 buttons. It can be awful. Where he looks at, over the past 40 plus years, yield increasing here, major grain crops, and pesticide sales here. If we actually look at the yield loss pre-farm gate and efficacy of control, this is loss from pathogens, virus, animal pests, and weeds. This is the potential loss, and this is the actual loss. So that's the gap between when insecticides are actually used. So the potential loss is around 18%. Pesticides reduce that 18% by almost 40%. If we combine pre-harvest with post-harvest, the loss of food and fiber, food for this one, is around 37%. So that's quite a lot. So we're not doing so great. And this, of course, really kind of is confronting with our increasing food demand and our global challenges, where we really need to reduce food and fiber loss, minimize degradation of ag landscapes, and reduce our farm inputs, including our insecticide use and pesticides more broadly. All of these goals and challenges are linked. None is more important than the other. And my argument is going to be that entomology has got to play an important role in that, and a much more important role than what we've done so far. Okay, so to me these key challenges, and the 2 messages from my talk today will be that, one, in order to do it, we have to connect the scales. So everything from the fine plant and plant-insect interactions all the way up to the landscape scale. And we also need to get much better at demonstrating impact. I thought some of the information on the ICRISAT website was terrific about the impact of the work you're doing. Okay, so arthropods. They're habitat linkers. The majority of these pests are highly mobile. They're moving through the landscape, and by their movements, they're connecting different patches, different life stages. They're spending their time in these different habitats. And so even though we spend a lot of our time trying to control a pest at the scale of the field, it's often very difficult because they're using far more habitats than just those fields. And what do I mean when I talk about connecting scales? Well, there are temporal and spatial scales. And population processes occur at various scales. So population processes are movement, so how insects arrive somewhere, birth, death, and how they leave. We know that at the scale of the plant, the fields, and the fields next to fields, and the farm, and the landscape, that a lot of our pests and a lot of our natural enemies are moving at these different spatial scales, including different time scales. In ecology now, it's well recognized that if we ignore these multiple scales, we are ignoring or missing patterns, and it means we are also lacking a lot of efficiencies in management. This is particularly true when we think about pests, and some of our greatest successes are in area-wide management. And that's because it often considers the scale and the population of the pests we're trying to control. All of this has to happen within this farming landscape where it's quite harsh environment, lots of disturbance. Insecticides are used to kill the pest, but we also are relying on beneficial insects to also, hopefully, suppress these populations. So quite a dynamic environment. And what do I mean by measures of impact? Well, a fairly kind of general or theoretical statement would be it must be linked to our management objectives, and the examples are many and varied. But they must provide evidence of a lesser problem, a greater savings, or that the science-based solution is actually being adopted. So I'd argue that in pest control, there's a lot of solutions that have been developed, but one of our greatest challenges still is getting those solutions adopted. Okay, so for the last 10 years at CSIRO, myself and the team that I work with, our research can be characterized by these three things: movement, landscapes, and how this is all integrated within a farming practice. So I think nowadays there's a kind of sexy, singular message that gets transmitted for silver bullets to solve these global challenges. We have situations of, being able, using a lot of technologies to produce large vegetables or pharmaceuticals or even trees that glow. It's hard for entomology to often compete with this, but these solutions, I'd argue, are not a magic bullet. And that, in fact, entomology and integration is going to have to be the key to work with some of these technologies once they get rolled out in the landscape, once we have humans interacting with these technologies. Okay, so for my talk, I'm going to spend the rest of the time on two examples that I hope address or start to meet some of these global challenges. The first is I'm going to talk about the link between perennial habitat and the agricultural landscape and crop pest control. I think this example will try to speak to this challenge of minimizing the degradation of ag landscapes. And then I'm going to look at the second example, moth behavior across cotton grain landscapes. Now I'm going to focus on two moths that are also global pests. Well, one of them is Helicobacter armigera, which you have probably know all too well here, and Helicobacter punctigera, which is the native bollworm in Australia. Now, this example, I think, tries to speak to reducing farm inputs and reducing food and fiber loss. Okay, so connecting scales. In 1999, a seminal paper by Carsten Tille and Nancy A Schellhorn looked at landscape structure and biological control in agroecosystems. They reported on some really fascinating findings. On the x-axis, they looked at the percent of non-crop area, and the y-axis, parasitism rates and damage. They used the oilseed rape system, pollen beetle, and a range of parasitoids that attack this pollen beetle. What they showed was this fascinating relationship that with more non-crop area, and in their case it was grasslands and woodlands, you got higher parasitism, but also you got reduced damage. This paper really spurred on a lot of scientists to conduct similar work around the globe. And so what's the global state of play after 15 years post this seminal paper? Well, there's approximately about 65 studies that have looked at this relationship between landscape metrics, so this could be the percent of land use, and abundance and diversity of natural enemies. So there's a positive trend. There's higher abundance and diversity of natural enemies with higher percent of non-crop habitat in the majority of those studies. Most studies actually focus more on the natural enemies that attack those pests, both abundance and diversity. A few even go as far as measuring pest suppression. And what these studies have done is demonstrate a clear link between spatial scales, so that there's a link between a field, a farm, and a landscape. So what's the problem, or the gap? Actually, there's no problem. It's been fantastic that this group spurred on this type of research that has really got a lot of people to think beyond the scale of the field. Well, there's a strong focus on natural enemy abundance and diversity in landscapes, but we need more examples of pest abundance in landscapes. So we still have very limited information on what's the relationship between the pests and some of these land use features in the landscape, such as the amount of agricultural landscape. Examples are coming. Most are in the northern hemisphere, but we need examples from Asia. We need more examples that integrate farming practices with landscape ecology studies. So most of these are studies that have been driven by the need and desire to conserve non-crop habitat and to conserve biodiversity. Several studies have demonstrated pest suppression, but suppression in a scientific sense does not translate to pest control, which is often determined by an economic injury level and something whereby a farmer cares about. So we need evidence of greater control, or greater savings, or evidence of strong impact. Okay, so let's look at this example of the link between perennial habitat and crop pest control. So of course, all my examples are going to come from the Australian system. So what's the problem in Australia? Well, the focus has been on pests and natural enemies in crops, and not other habitats in the landscape. So this is some of our work that was funded by the grains industry, and this is a landscape in Brisbane, New South Wales, across that landscape. And what we did is we asked a very simple question: where are the pests of grains and predators found? So in grain systems, we have a pretty good idea about who the pests are, and we have a pretty good idea about which predators attack those pests. So we did extensive sampling of a lot of the key predators, both the adult and the juvenile stages, as well as pests, adult and juvenile stages. And we did both of those because juvenile stages tell us where reproduction is happening, because most of these juvenile stages are flightless. So they're usually associated then with a plant, in the case of pests. So we sampled in 3 different regions throughout Australia. So the subtropical, the lower temperate, and then the Mediterranean of WA around Albany. 6 different landscapes with over 500 sites embedded within each of those landscapes for every month for 2 years. And we sampled from native vegetation, we sampled from weeds, and in those native veg samples, we sampled a crop nearby and a crop far. So lots of sites, 2 years monthly. So what do we find? Well, here's some very broad course data. Now, the reason it's so coarse is that farmers are often not dealing with just one pest. They're dealing with a range of pests. So I'm lumping these together to look at broad general patterns. Pest is in the red, predators in the blue. This is crops, pasture, and native veg. These are the different from the sites and the landscapes of New South Wales, Western Australia, and Queensland. And the first thing that really should pop up is this really strong pest signal in pastures. In crops, there's not much difference going on. There's a few higher predators in crops in Queensland. In native vegetation, there's a lot more predators hanging out than there are pests. But this is by far the signal that jumps out the most. Within native remnants on farms, they can often be in various conditions. So you can have a lot of native understory and midstory and overstory, or it can be overgrazed and you can end up with a lot of weeds. So within those remnants, we sampled from a range of different plants. We sampled from mixed grasses, native plants, and weeds. What you should see here is that on our weeds, again, we had lots of pests of grain crops. From our native plants, we hardly had any pests, and mostly our predators of grain pests. And in our mixed grasses, we did have some pests. So that was a good coarse picture. If we just narrow down and look at one pest that's often a sporadic pest, it's a native insect of Australia, Nancy A Schellhorn, the rudderman bug, here's the adults and the nymphs, we can start to look at their pattern throughout the year and how they move. So this is January to March. The pasture sites are in blue, and this is rudderman bug density. The darker the color, the more dense the populations. And you can see that where we sampled, you can see that rudderman populations are present in these pastures. So now, April to June, wheat, barley, canola are going in the ground. So that's what these are. These are all the sites for wheat, barley, canola. And you can also see in pasture the rudderman populations are building. Now you can see how they're moving over into the crops. So the ones that aren't labeled are wheat or barley. And then at the end of the season, still you can see we've got that pest that's in those crops. So here's a good example of how the seasonality of the landscape and the cropping systems, that the rudderman bug's actually taking advantage of that. But it's not just pasture. Here's the weed percentage, pardon me, in the pastures on the x-axis, and the rudderman juvenile populations on the y-axis. And more weeds in pastures equals more rudderman bug juveniles. So pastures in good condition are not the problem. Weedy pastures are the problem. And those are often the ones that have been over-fertilized or over-grazed, and then you end up with broadleaf weeds. Okay, so what about some of our beneficial insects? And when are they there? Well, the brown lacewing, Micromis tasmanica, is a voracious aphid predator. It's the adult, and that's the juvenile. These two graphs show the sampling for every month for 2 years. We let people have a break here. That's what that gap is. We let them have Christmas. So this is the symbol for adults and juveniles, and this is in the native veg. These are in the crops. And what you can see is that both the adults and the juveniles are using the native veg. And here the population is declining, and you can see it increasing in our canola. Canola or wheat? That's a wheat, sorry. You can also see how we have juveniles increasing in the canola. So this habitat is really important. It extends, here's the cropping season, but they're in that native veg for longer in the period of the cropping season. Some of these organisms are quite long-lived. And for example, lady beetles can live up to 1.5 years, some of them. Most of them live for about 4 or 5 months. But certainly to maintain their life cycle, they need these alternative habitats in the landscape to be present to then colonize crops. Okay, but what does this mean for actually pest suppression in our crops? Well, there's been a lot of theory that's looked at the early arrival of predators and what it means for pest suppression. So this is time on the x-axis and pest density, and this is a classic exponential increase, particularly things like aphids, mites, roots. If we add a biocontrol agent here, we get a bit of suppression happening. But we know from theory, if we add it early in the piece, we get quite good suppression. So we set out to test this hypothesis in our landscapes using some experimental approaches, not just observational approaches. And we asked the question, is pest suppression greater in fields, near remnant veg patches, compared to far? Now, our approach is not one that I necessarily recommend to people because it was crazy. We used thousands of sentinel plants, some which were clean and some which were infested with different life stages of pests. So for example, for cotton, we put Helicoverpa egg cards on them, or we infested the plants with whitefly nymphs. So that would be a sentinel that we could put out, and we put it out on bare soil that was mimicking a crop just coming out in the ground. We placed them in grids, so we placed sentinel plants in the native remnant, 25 meters away, so in a crop field adjacent, but it was bare, right? So these were the only plants that were there, or 400 meters away. And we placed these grids all throughout landscapes and replicated landscapes. We also conducted a similar study in the Lockyer Valley, which is the salad bowl of Australia. And this time we used melon seedlings and melon aphid, which were melon aphids on cotton. It's called cotton aphid. And again, we had different treatments of exclosures, of open sham cages with these aphids on there. And these were replicated, 19 fields throughout the Lockyer Valley, and each of these fields varied in terms of how simple or complex the surrounding landscape was. This is an example of a complex landscape. So a slightly different design, but again, to ask the question, and these were put out on bare soil, what's the role of the landscape in contributing predators to suppress these pests? So here's some of the results for cotton. Now all the cotton ones were only out for 72 hours. And so what you can see is this is parasitoids per plant here. This is parasitism in the remnant, near field, and far. And you get a stronger trend within 72 hours of parasitism in the remnant and near than you do far. We did it at 3 time periods. But what you can see is how variable that response is. So from our cotton example, we'd say that this ecosystem service of pest control is spatially and temporally erratic. So it's not high and it's not consistent. From our melon example in the Lockyer Valley, to make a long story short, by far the best predictor, and one with a fairly significant R squared, at least for ecology and these kind of studies it is, the more percent of lucerne you had at 1.5 kilometers, the greater your pest suppression. So this is the amount of lucerne surrounding each of those experimental fields where those sentinel plants were put out. And this is the surviving aphids. So predation was greater when there was a lot of lucerne around that 10% at a scale of 1.5 kilometers. We also put out a lot of sticky cards, non-attractive, in these different habitats, and not just look at the pattern or the predictor of land use to pest suppression, but also the density of predators available. And again, we see a somewhat similar trend. In lucerne, we got better pest suppression, but at the scale of 500 meters. So in lucerne crops, where there were predators available, that's where we were getting the best pest suppression. But also other crops at that same scale. So in this study, native vegetation in remnants was not playing the strongest pest control function, but a perennial crop such as lucerne and other crops were. Okay, so for this first example, a snapshot of the summary. The first important message would be that we have to think beyond the crop, that the landscape context matters. So I showed you that native vegetation clearly provides habitat for beneficials, and beneficial insects being natural enemies, especially important out of the cropping season. Weeds harbor pests. They can also harbor some beneficials, and I'll show you that in a minute. Perennial pasture and lucerne plays a key role in landscape for beneficials and for pests if pasture is degraded or weedy. And so what we have now is a list of all these plants that we've been sampling these pests and beneficials from. Anything to the left is a plant that is highly pest dominated. So these are samples over, hundreds of samples over 2 years. Anything to the right is predator dominated. And the color pattern, what it should show you is that weeds all sit up here. These are the crops in the middle. This is pasture. And these are all native plants. So what this is allowing us to do now is we've created a range of fact sheets for rebed options such as windbreaks or shade and shelter when people have mixed cropping systems of livestock and crops that growers can actually use for revegetation activities or for restoration. If there's ways in which water courses or riparian areas need to be restored, then we can link some of these plants for ecosystem services beyond just the intended purpose of restoration, but also to capture the ecosystem service pest control. But these examples of pest suppression are not examples of pest control. And that's what we're currently working on. It's a project where in these cotton and grain landscapes, if these types of perennial habitats are really having an impact, then we would expect pest populations to stay lower for longer. We would expect pest populations to not go above threshold as fast as when landscapes are in quite homogeneous or highly intensified areas. So that's the question we're working on now. Okay, so summary for example 1, I showed you how mobile insects in the landscape are actually linked at different spatial scales, that there's some theory that predicts why this can be beneficial for ecosystem services, and that we have some knowledge about these perennial habitats playing an important role. Have we demonstrated impact yet? I think not, so we need to work towards it. What we are doing is linking conservation biology and agricultural production, because it's a key component of the system. Okay, let's go to example 2. So this one is Helicobacter species and moth behavior across the landscape. Armidra, a global pest. Again, we spend a lot of money on it, and a lot of people have bought houses and bought cars and put children through school on this pest problem. So it continues to outsmart us. It has a tremendous host range. In Australia alone, recorded on 159 host species. So for both species, Armidra and Punctidra, there's about 85 species. Additional 34 of those are for Armidra. Armidra likes grasses. Punctidra does not. So in Australia, we have BT cotton, like you do here in India. And now about 95% of all cotton is BT cotton. The impact of BT cotton has been tremendous in terms of insecticide reductions. So when the first generation of BT cotton came out, we saw a 44% in insecticide active ingredient going into the environment. With the second generation of BT cotton, we've got about an 85% reduction in active ingredient going into the environment. So it's had tremendous impact. However, the reduction in insecticide use really depends on continued effective resistance management plan and also biological control for other helicoverpa susceptible crops across the landscape. So what I mean by that is here's a landscape of Darling Downs in Australia, and most of these crops that are planted out are susceptible to helicoverpa. So it's a virtual smorgasbord, not unlike what happens here in India. So the work I'm going to talk about takes place there, and there's numerous sites in these big 20-kilometer landscapes that run along the course of this waterway. So for BT cotton, for helicoverpa, it's a population sink. Eggs are laid and they die. You don't get offspring, or you shouldn't, as long as the technology is working well. For sorghum, which is the other dominant and often most dominant crop in the landscape, it's a source. But it's only a source for Armidra, not Punctidra. Punctidra hates grasses. It's probably a good thing. Well, actually it's not, but that's another story. And then there's also pigeon pea, which for this technology, this is the mandated refuge. This is the only mandated refuge that can be used in addition to conventional cotton. So no other crops can now be used as a refuge. So it's a source for both. Now this refuge strategy means that growers have to put in 5% pigeon pea or 10% unsprayed conventional cotton within 2 kilometers. They also have to do things like planting windows and pupae busting. This is work that's been modeled to show that it's the best way to keep resistance at bay. But the real issue is that it doesn't cost that much to plant the seeds for pigeon pea, but there's a lost opportunity cost because you're not able to plant cotton. So there's a constant drive in the industry to try to make it more efficient, the refuge management strategy, but also make it effective. But the curious thing about Armidra is even though it's a global pest and people have been studying it for a long time, we really don't have a great understanding about how this moth interacts with the landscape, with the crops of the landscape. So the hypothesis I want to talk about is one about we would expect helicoverpa A density to decline as huge patches of BT cotton, as cotton increases. And the reason for this is that it's a sink. Moths keep laying their egg in there, but you're not getting any recruitment. So all the eggs that end up there have to come from the outside surrounding landscape. As the crop gets bigger, you would imagine that becomes more difficult. Our second hypothesis for Armidra only is that we would expect increased eggs with the increased amount of sorghum. Armidra loves sorghum. The more sorghum, you would imagine the population is able to take over. So pretty simple hypotheses. And what we did is in these different landscapes, we selected fields that varied in size on a gradient. So for example, one field, our first field would be selected as a minimum size of 185 hectares, and then that reduces. So we did this for cotton for 5 years. We did it for sorghum in the last 3 years. And for every one of our sample fields then, we characterized the surrounding landscape. There was 26 different land use categories, and they were mapped for all the years across all the landscape. The metric I'm going to talk about is just helicoverpa eggs. That's it right there. Eggs tell us about female moth behavior. Eggs in BT cotton tell us about colonization, because you shouldn't be getting recruitment. So we counted something like 23,000 eggs. We collected a subset of those. We could also talk about their fates. We knew how many were parasitized. We could rear them out so we knew what proportion were Armidra, what proportion were Punctidra. And this is kind of a summary of the data we have. Now, in Australia, I don't know what a normal year is anymore, because our 5-year data set, the first year was a drought, the second one was a big flood. The third year was regular or normal. The fourth year was a flood, and the end of 13-14 started out great, finished as a drought. All the sorghum crops were lost pretty much. A lot of them got sprayed out. So it's a 5-year data set with numerous sites, 72 fields in each of the landscapes. Is that right? No, 24 in each of the landscapes, 72 altogether. 3 landscapes, and these represent the different crops. So for our egg data, this is what it looks like over time. Now the reason it looks like this is because funding became available. We increased our sampling and we changed our questions slightly. And then we also have a lot of data on moths, pheromone traps, UV solar lights that we made, and pupae digging, so we can find out the eggs, what goes in and what comes out. For the purpose of this talk, I'm just going to focus on this egg data and moth behavior. And to give you an idea of how changing the Darling Downs landscape can be, this is the different years, and this is BT cotton is in green, the source, so those would be other crops that are also sources for armidra, and this would be unsuitable land. And you can just see that in some years, you only have 13% BT cotton, where in other years, you have close to 50. So the shrinking and swelling based on water availability and market price also has, can really change this population dynamic. And something only if people are interested in, it has huge implications for resistance management, but I won't touch on that in this talk. And to give you an example, the first piece of data, armidra is in the black, punctidra is in the gray. This is the percent of those eggs collected. This is just a proportion of each for each of the trips in the different landscapes. In the drought years, we see a higher proportion of punctidra. It's as if it's saying, oh, I don't like cotton, but if it's around, it's the only thing around, I'll lay my eggs there. In flood years, didn't see so much, hardly at all. Normal years, we see the classic pattern where punctidra shows up first and leaves, and then armidra dominates. And then in this last drought year, again, you see that punctidra is dominating. So the way in which the landscape is changing, there's also a changing of the 2 pests. Okay, so having a 5-year data set of eggs across a large space and time creates some complexities for analysis, but it's also an enormous opportunity. So here's what we're doing, and here's the novel part of our work, the part that might make crop protection more interesting. So we're using these random forest models. So these are machine learning techniques where you have lots of data, both spatial and temporal data, that basically goes into these random subsets and it creates these trees. So it takes the average of a large series of decoupled data and it runs 1,000 iterations to say what are the key factors that are explaining this variation in our response variable, which is egg abundance. So there's lots of features about random forest models that make this great. It's distribution free, large number of predictors of different types. You can use both continuous and categorical. Handles missing data, handles collinearity, and in landscapes, compositional data. There's nested spatial scales. But it's a non-parametric modeling approach. But for us, we had 4 spatial scales around each of our fields, 26 land uses, and temporal variables. So there was 100 variables. So this was one of those questions where we decided to throw up all the variables and to see what happens, what comes out in this kind of approach. So here's an example of what we get. So here are the top predictors from the random forest and what's called a proved regression tree for armidra. And what it does is it starts to make distinctions between where you have different breaks. So for example, in this data set we can see that there are some differences in years. And in terms of the largest number predicted helicoverp armidra eggs, when there's a little bit of sorghum, only a little bit of sorghum around, you're getting more eggs in BT cotton. You're also getting more armidra around when there's more punctidra around. In years 1, 4, and 5, we see a bit of a landscape effect. Fallow land and a particular landscape is predicting most of the variation. And in terms of our smallest number of eggs, well, prior to cotton flowering, that's when you get the least. And then there are some other spatial factors that come in as well for armidra. If we look at punctidra, you see that the tree is much simpler. Fewer things are really starting to say what explains punctidra eggs going into cotton. And most of these are temporal variables. Flowering cotton, moon phase. Now that one I warming too. But now I have, and I'll show you why in a minute, different years and an estimated number of Helicoverpa armigera eggs. So when there's more armigera around, there's also more punctigera around. Now what we're doing, so there's the year, and so the largest predictor is, yes, this was the most surprising result. The largest number of predicted punctigera eggs is when there's a waxing moon of greater than 80%. So from 80% to full moon, more punctigera eggs. Also cotton flowering. So what we do with this random forest approach with these 100 predictors is that we take these top 12 predictors and we then move it into a parametric model. So this is a way that we can combine space and time to then move from an explanatory tool where we have 100 predictors to one that we're really interested in predicting the abundance of eggs. And by moving into a parametric model, we can increase our confidence and we can also ask different questions. So this is the result from the parametric model after going through this process. And I'm trying to simplify it. So if you remember, this was our hypothesis. But that's not what we get. In fact, we get exactly the opposite. So from our parametric approach, where we used a general linear mixed model, the main factors that predict the abundance of Helicoverpa armigera eggs going into cotton are really two things. The percent of cotton at 2 kilometers, so the bigger cotton is, the more eggs you get, and we see a local effect. So some landscapes, so Nandi has fewer eggs than Cecil Plains, and Pampas always has more eggs than Cecil Plains. Those are the two consistent factors. And we actually have a very good fit of an ecological model that deals with big space and time. So not what our hypothesis was at all. Now we're just starting our analysis for armigera eggs and moths in sorghum. In our early descriptive results, this is a schematic representing that. If you have sorghum that has a lot of Bt cotton around, compared to sorghum that has more sorghum around, you get about 20 times more eggs in sorghum surrounded by Bt cotton than in sorghum in the sea of sorghum. This is suggesting that there's some preference, that armigera has preference for different crops, but the landscape context and the way it responds really changes the behavior of putting eggs into those crops. So basically initiating the pest problem. So what about for punctigera? Again, that was our hypothesis. This is the species that doesn't like grass. These are the key predictors, and again, quite a good fit for our model. So the first one is, and this again, this is a simplification instead of showing you tables with numbers. But the first one is the percent of a full moon. It comes out very strong. This is a big surprise to me. It made me even think we should change the name of our group to like crystals, moons, and I don't know, incense. I'm not sure. But it's a very strong predictor. Anything from an 80% waxing moon to a full moon really can predict the abundance of punctigera eggs going into a crop. Also cotton development. From squaring to flowering, more eggs. And the amount of fallow or bare soil at 1 kilometer. If a cotton crop has a lot of bare soil around, punctigera is more likely to stay in that crop and lay eggs. So, nope, our hypothesis based on our biology and what we think was happening is not happening. So for the summary of those results, I think first of all what it's done is here's a global pest and its cousin, punctigera. And this type of approach where we're looking at large spatial scales and we're including space and time into these different modeling approaches is helping us understand these key drivers. Drivers that we haven't seen before. So spatial drivers are dominating for armigera. So again, there's a local landscape factor. So something's happening in Pampas that always makes it have more Helicoverpa armigera eggs than it does in the other one. And there seems to be this strong preference that armigera is queuing into, and it has sorghum over cotton. And so again, as we think about the configuration of the landscape and the spatial arrangement, we can start to see that we would expect a different dynamic in terms of the pest population going in. Temporal drivers dominate punctigera, 80% waxing to a full moon, cotton development, and there's no crop around that Bt cotton. Now others have made statements about the moon and moths. The influence of the moon is likely to be complex. I think true. If you talk to farmers, they will often say that they notice things at the full moon, lots of activity. You look at vertebrate species have enormous eyes. Maybe they really need this, they're nocturnal. I don't know. It's a tricky result. But to summarize for example 2, so I showed you that Armidra and Punctidra, when we start to think about these are real landscapes and how these moths interact and behave, we can really see that there are some spatial and temporal drivers. Now, we haven't demonstrated impact yet because we're just starting this analysis. But what we're trying to do is feed this into the resistance management plan, both making it more efficient and also feeding it into our resistance evolution models to make sure that we're delaying resistance as best as possible with the way in which we deploy a resistance management strategy across the landscape. And ultimately, we hope that we're contributing to these global goals of minimizing the amount of pesticide going into the environment while minimizing the loss of food and fiber. Okay, so I want to finish with just some fun stories about future opportunities to reduce farm impact. So I know at ICRSAT and a lot of agronomic language we talk about sustainable intensification. And I'm always fascinated by the way in which terminology gets developed in different disciplines. Well, ecological intensification is a terminology that's being used quite heavily in the ecology and conservation literature. So I just want to finish with 3 quick fun examples that I think are interesting for the future. One is some work by a colleague, Ricardo Bamarco, where they've been managing pollination in crops to reduce the amount of nitrogen that goes in. So a replacement idea. So nitrogen is expected to be increased in cost, and if we get better pollination management, then you potentially can get exactly the same yields without the amount of nitrogen. There's also Olga Lundin's been doing some interesting work to show that on plants where you get good biological control and you combine them with pollination, you get fantastic yield. Either of those alone, you don't get the same outcome. And one of my personal favorites that we were talking about at lunch, but clearly is only appropriate in particular kinds of soils. This is some work by Theo Evans. And in the marginal land of Western Australia, they showed that by not using broad-spectrum insecticide on the soil at the beginning of the season, which is very common in grain production in Australia, you maintain termite and ant tunnels. These tunnels, they showed, demonstrated mechanistically, allowed for increased water infiltration and nitrogen uptake. And it resulted in yields of increase of wheat from 37%, which is phenomenal. This work was published in Nature Communications. It's these types of ecological intensification, particularly as we're trying to reduce our insecticide loads, that we really need to start thinking about. Okay, so in conclusion, I've shown you examples on movement, landscapes, and farming practices, and how we hope that these can start to contribute to some of our big global challenges. In terms of impact, well, I think the capture of pest control services from landscape mosaics is pretty important, and it also is a means that we have to think about protecting the natural farm assets. And secondly, the potential to refine resistance management plans can contribute to that global challenge of reduced food and fiber loss and reduced pesticide use. So back in 2013, Science had a special issue called Smarter Pest Control. And with the 15 to 20-fold increase in the amount of pesticides used worldwide in grains, it's something that we really need to be thinking about. With that, I'd like to thank my collaborators. I get to stand up and talk about we. Well, we all know who the we is. So these are the team in Brisbane. Thank the funding agencies and also many growers who open up their properties for us. And this is from the cotton example. So with that, thank you very much for your attention, and I'm happy to take questions.
Hey Nancy, that's fantastic examples from Australia. Actually, really keen to learn more about some of the work you're doing there. But anyway, I'm going to throw it open. I think I've mentioned that we have a new Director General. He started his career as an entomologist and then floated with maize breeding. So I'd be interested in his view on, how do you deal with pests in the landscape from a molecular biology technology approach or an ecological approach. So David, I'm not going to throw to you immediately, but you might think about it. So any questions?
Throw it to me directly then. Thank you, that was great. My question actually is focused on a summary slide that you were talking about termites and ants. And and the increase in yield. In a way, it surprises me given the competition for carbon and its integration into the soil that especially termites pose. So I wonder, if you could offer some insight into this, because there are trade-off ecological benefits of especially termites. And then to your question, Peter, yeah, I think the integration across different scales is very important. You're working on the landscape scale. Harry's working at the molecular and biochemical level as it relates to host plant resistance. Clearly, we need to bring all of these together, as you point out, to deliver integrated pest management. And I guess our challenge is how do we bring all these disciplines together? And it really, I think, comes down to knowledge exchange and building the kinds of teams to allow that interdisciplinary approach to technology development. It'd be great to gain your insights on how you've cultivated those collaborations with people like Hari that are focused on the host plant resistance side, and where you're running into challenges in facilitating that more integrated approach for integrated pest management. So 2 questions.
Yeah, so I'm not an expert on soil invertebrates. I really like the example because I think it's so understudied. Certainly in Australia, I think that it's so rare that we actually even get to look at this question because the predominant pesticide practice is to start the season with a broad spectrum insecticide. So I think this is the first glimpse. At lunch, in talking to Anthony and we were talking about the challenges and the difficulties with it and what it meant for when it starts to rain. I don't have that knowledge to say when it's not going to work. But I think these are the kind of examples that we need to be thinking more of. No-till systems in Australia now, what we're seeing is a whole new emerging pest fauna. That's because you're getting more organic matter in the soil, but you also are putting down broad spectrums all the time. It's the same thing with diamondback moth and canola in Australia is a problem. It's not in South Africa. Now, it's exactly the same climate factors, and they have diamondback moth there, but they don't use any pesticides on their crops. So what happens often is you get this mass influx of diamondback moth into canola that you've already taken out all these predators because of the broad spectrum use early on. So it goes against what we know about best practice. So I think people who know about agronomy and soils are more able to speak about the potential opportunities with this than I am. That's the first question. I welcome that discussion. I believe the second question was more about how you go about integrating. Well, in talking with Tara's group today, I said to them, we've done a really good job at completely ignoring host plant resistance at the moment because we're looking at these broad scales. And I think our next big challenge is to start to now, as we figure out methodological ways to think about populations across space and time, how do we now connect it with that variation that host plant resistance is going to give us, and hopefully that additional suppression of pests. So I think that's our challenge, and I think after the headaches of scaling up and what it means, how challenging it is, I think our team welcomes the opportunity to start to scale down a bit and start to make that link.
Okay, answer one is probably there is a lot of data on its beneficiaries and of course the harmful ones on the Darling Downs right from the days when transgenic horticulture was introduced. So maybe a meta-analysis of such data, which is probably in the Cotton Research Corporation's annual reports, would be very useful. Another thing probably to look at is which of the crops serve as a source or sink for the pests and the beneficiaries. That will lead us to identify crops, crop combinations, and or mosaics that will then lead to the pest suppression. One result that surprised me was that there were more herbivores in the pastures than the carnivores. So I would have expected it to be otherwise. So we're using that.
Yeah. So I think in Australia, exotic weeds are a relatively new introduction, and a lot of the pests are also exotics. So I think there's some host plant links there already, and that's part of the reason. And so they're just able to get going. Whereas on the native plants, a lot of these come from Myrtaceae. We see a lot of our beneficials, our predators hanging out there, our coccinellas, our lacewings, our native coccinellas with Cryptolemus, which you don't see, I don't think a lot of the introduced pests have managed to really feed on their safety. So I think that's why the ratio is so different, and why weeds, broadleaf weeds in pasture, where they're never controlled as well. I like your suggestion about a meta-analysis. I think you and I talked earlier about, we have this long-term data set about Helicoverpa, and we can see now over the past about 10 years, the populations have declined. One hypothesis is due to BT cotton. Another hypothesis is due to the tremendous reduction in SPs and OPs. So we are, seeing these broad-scale patterns in terms of pest suppression. But, the meta-analysis we'll have to think about more.
Nancy, from me, CSIRO has got a number of changes in its organisation, one in which a matrix like CG, but another experiment was that some time back they put agronomists like me in the same organisation as wildlife and ecology people, ecologists, as an experiment. That was weird because the whole idea of remnants in a landscape for me was just a waste of agricultural land. And so working with Nancy and others, I realised that those remnants, native vegetation, have important ecosystem services. And so that's a real learning, and I think that most in agriculture, certainly in my education, didn't realise. So it's nice to hear stories like yours that can quantify what the benefit of a remnant is. So I'll let you expand on that, but I'm also going to throw to we have the director of water, land and ecosystems in the CGIAR in the room here, and I'm interested to know how much work has been done in the CGIAR about this idea of landscape ecology, not just water, but also these ecosystem services beyond it. So first you, Nancy, then to Andrew.
Yeah, so because we're talking about arthropods, that's the focus. But I also, so if a remnant is weedy, I'd say it becomes a problem. But also, I'm not suggesting it can just be locked up. It can be a habitat for feral pigs if it's not managed. And so it's not just something that has to sit there and is pristine. But if it's managed, I think it does have an enormous, positive role to play.
One was about the pollination.
Yeah.
Andrew?
Yeah, thanks. And that was great, Nancy. And I certainly think from the WLE perspective, what you're saying resonates very well. So within the program, we actually do have people working at that scale. And I can give you some examples from Costa Rica, where we're looking at what we would term natural vegetation barriers, effectively to control some of the pests associated with coffee production that have worked extremely well. And the way we've been working with farmers and also utilities people, particularly hydropower companies, effectively to look at structuring these mosaics across the landscape that has benefits from a hydropower perspective in that it reduces sedimentation, particularly riparian vegetation, sediments going into the storage structure. And the second one is looking at farmers creating what would be termed greater diversity in their landscapes with natural vegetation that has very positive impacts on their coffee production. Another example, and this is from Asia, and it's work that IFPRI have been doing with entomologists in North China. And there they've been looking at landscape structures that would result in benefit, you could say positive benefits with respect to cotton production, and specifically looking at ladybirds and the role that ladybirds have in cotton systems. And what they've been finding is that it's not the diversity that's critical in many of these systems in northern China. It's a combination of diversity and crop rotations. So if you're looking at farmers that have a wheat maize rotation, they tend to have higher levels of ladybirds presence in their subsequent cotton crop. And the key element to that is actually the maize crop, because effectively they don't use insecticides in their maize system. So you know, a lot of what you're saying has tremendous resonance with us, and we strongly believe that a mosaic or contrasting structured landscape has very significant benefits to producers.
Thanks, Andrew. Any other last questions? Okay, Nancy, I thought that was a fantastic seminar showing some of the diverse work that you and your team are doing in Australia. And I can say too, from my knowledge, so she talked about a region that I've spent the last 25 years living in, Pampas and Dalby and places like that. And the farmers there take notice of this work. And it's really a key that you've got farmers interested in landscape ecology, not just how do I manage my sorghum crop or my cotton crop. And so that's a real credit to you and the team and the work that's been done in that region. So join me in thanking Nancy for the fantastic seminar.